Hello, my name is Valentina Grisanti. I am a graduate student in the lab of Dr. Zubeida Cruz Monserrate. Today I will present my abstract titled Increased Physical Activity Delays Development of Obesity-Induced Pancreatic Ductal Adenocarcinoma in Mice and Modulates Inflammation. Obesity increases the risk to develop pancreatic ductal adenocarcinoma and also increases inflammation independently of PDAC, which can modulate this disease and worsen its outcomes. We hypothesized that implementing lifestyle interventions that decrease obesity by modulating fat consumption and increasing physical activity could be used as preventive mechanisms against PDAC. To test this hypothesis, we exposed obesity-induced, genetically engineered PDAC mice and control mice to a high-fat diet before randomizing them into 3 different interventions. One group continued on the high-fat diet, another group changed to the control diet, and the last group was exposed to both a control diet and physical activity. The mice in the controlled diets and physical activity interventions gained significantly less weight overall as seen in Figure B. Figure C shows that all the mice in the high-fat diet control intervention developed PDAC, while only 70% of the mice in the controlled diet and 40% of the mice in the controlled diet and physical activity interventions developed PDAC. The fibrosis and inflammation scores, while not significant, showed a decreasing trend in the groups that changed to the controlled diet and physical activity. Next, we explore the role of physical activity and diet in obese mice before the onset of PDAC. We used C57 black cis mice that were given a high-fat diet and randomized into different interventions of diet and physical activity prior to orthotopic implantation of PDAC tumor cells. Only the mice that underwent the controlled diet despite the physical activity showed a significant delay in weight gain as seen in Figure B. However, the tumor weights for all the groups remained similar, suggesting that neither physical activity nor diet-induced weight loss prevented PDAC tumor growth in this model. In another experiment shown here, we found that physical activity in the absence of obesity did not modulate tumor growth or chemotherapy outcomes in an orthotopic mouse model, but in Figure B, you can see that the group that in the physical activity combined with gemcitabine chemotherapy increased the percentage of splenic CD8 T cells significantly. We also measure inflammatory cytokines in the blood of obesity-induced PDAC mice and overweight human subjects that underwent a physical activity intervention. Figure A shows how the PDAC mice that did not do physical activity displayed increased levels of inflammatory cytokines over time, compared to the PDAC mice in the physical activity intervention. And Figure B shows a decrease in the inflammatory cytokines after the intervention from the human subjects. To conclude, physical activity and diet-induced weight loss delay the progression of obesity-induced PDAC in genetically engineered mice and reduce inflammation, but this is not seen in the orthotopic mouse models. Further research should aim to determine the mechanisms by which physical activity might prevent tumor progression and whether it could reduce the incidence of PDAC in high-risk obese individuals. Thank you. Here are my acknowledgments and contact information.